welcome to everybody. Thank you for attending this GVN webinar. And as usual, before introducing Dr. Nogueira, I just want to make a few points on the Global Virus Network for those of you who do not know it. So this is really a science-driven and independent, and these are keywords, networks created in 2011 by Bob Gallo, Billy Hall, and several other colleagues. And the GVN is now including about 68 research centers worldwide and 12 affiliate centers. And our activities merge research, in particular with the GVN task force, education, training, very important with the GVN Academy program, and advocacy and communication. And you have all the details, obviously, on the website. Relevant to the presentation from Dr. Nogueira is the creation of the recent and ongoing creation of a GVN task force on dengue virus. So in this context, the GVN webinars are a very important component of our activity. And it's a real pleasure to welcome Dr. Mauricio Nogueira. Briefly, he is a professor in microbiology and infectious disease at the School of Medicine of Rio Preto in Brazil. He's an MD, PhD, trained in the University of Minas Gerais, and he had a postdoc in the NIAID. And this is a well-recognized and highly recognized international expert focused on arbovirus research. And he has been also the former president of the Brazilian Society for Virology. He's a member of the American Committee for Arthropod-borne and Zoonotic virus. And he's an associate adjunct professor at the Department of Pathology, University of Texas Medical Branch. Dr. Nogueira has always been very supportive of the GVN and has attended several meetings, and it's really a pleasure to welcome him for this lecture. Thank you very much, Mauricio.
Thank you, Dr. Brechot. It's always a pleasure to collaborate with GVN, and especially now also in the task force, and it'll be my pleasure to work. I'd like to thanks for this invitation, the opportunity to speak to this very selected network of scientists. So just I have a quick question. Are you seeing my screen, I guess? So I'm gonna talk a little bit today. We're gonna revisit the interaction of dengue and Zika. We're gonna try to show new data showing that we have a very complex immune interplay between these two viruses. As has been said, my lab has been working with arbovirus. That means arthropod-borne virus, that are viruses that were transmitted by arthropods, usually mosquitoes, but can be ticks also. And most of these arbovirus are in three families, the Togaviridae, Brunaviridae, and Flaviviridae. In the Flavivirus genus, we have several important viruses like dengue, yellow fever, and other ones. And dengue, yellow fever, and some of other viruses from other families are responsible for about 95% of the arbovirus disease in Brazil. We're going to focus today in the flavivirus and especially the dengue virus. As everybody knows here, dengue virus is a single-strand RNA virus in a positive polarity and codifies several proteins. It's a small virus with a small genome, but has a very complex interaction with both mosquitoes and the humans that are affected. Now we have, has been described 4 serotypes of dengue. Among these serotypes, we have about 65% of genomic similarity. Within the serotypes, you have several genotypes that have about 6% genomic divergence. This similarity among the serotypes gave us some variability that in the immune response. After an infection by one serotype, we have a very strong specific immune response to that serotype and some immune response without long-term cross-protection between serotypes. We have a short-term protection, but not a long-term protection among the serotypes. We also have genotypes inside these serotypes, and these genotypes have about 6% genomic divergence among them, and they lead us to difference in antigenicity, infectivity, and tropism among the genotypes. Dengue in Brazil has been a very important public health over the last almost 40 years, with number of cases being increasing year by year in a trend that leads to much more cases and much more deaths over the years. The Aedes aegypti mosquito was extinct in Brazil, but we have a reinvasion of the vector about 1976. We have the introduction of dengue 1, first 82 and then 86 and 90, giving our first dengue outbreaks in the modern area, followed later for the beginning of hyperendemicity in Brazil, when we have a dengue 2 introduction in about 1990. And then we have two serotypes circulating simultaneously in Brazil. By early 2000, we have the introduction of the dengue 3 in Brazil, and we begin to see sporadic cases of severe dengue, then called at that time dengue hemorrhagic fever or shock syndrome linked to dengue. They are now classified as severe dengue by WHO, and we use this classification in Brazil. And after 2010, we have the introduction of the fourth serotype, and the severe dengue, especially the dengue hemorrhagic fever and other forms, became a serious public health problem in Brazil. Also, in the last 10 years, we saw another introduction of flavivirus in Brazil that was the Zika virus. The Zika virus is morphologically indistinct from dengue. It's also a flavivirus. He has a very similar genomic organization, but has about 58% genomic similarity in their envelope protein are close but not equal. So they have some similarity, they have some common epitopes, but not all of them, of course. And what we are seeing now in the world is increasing range of the Aedes aegypti mosquitoes. We have an increasing report of dengue in these regions between the tropics, but we still also have some cases in Japan sporadically and South Florida. And also we have, I can say, sustainable transmission of Zika since 2016, 17 in Brazil. We also have Zika in most of South America. We have some endemic transmission in Africa and some transmission in the Pacific ring. So dengue in this other flavivirus has a very interesting interaction with the immune system. All flavivirus infection induces a potent expression of neutralizing antibodies for that specific virus. In these antibodies induces a partial and temporarily reduced cross-protection between serotypes. And this reduction of protection is also linked to some more complex interactions with this immune system, especially the antibody-dependent enhancement. That we're going to talk in the next slide a little bit more. But we cannot forget also that this flavivirus induces also a strong T-cell response, and heterotypic infections for different serotypes of dengue, for example, also can induce unbalanced T-cell response that leads to a phenomenon called original antigenic sin. But in fact, what we have that this immune interaction between different infections or subsequent infections may modulate protection, disease severity, and even the epidemiological fitness of different strains of virus. So the antibody-dependent enhancement, it's a phenomenon that's really discussed in the dengue field. I choose this example by Steve Whitehead that I saw that was in the audience now. That shows that when you have a heterotypic infection from dengue, for dengue infection, heterotypic antibodies, instead of neutralize, will facilitate the infections of monocytes or other FC expressing cells that leads to an increase of viral load and increase of disease. But this is a very simple model that shows only the antibody-dependent enhancement, but the situation is much more complex. We have, of course, this antibody-dependent enhancement of replication mediated by this heterotypic infection, but we also have what is called the original antigenic sin. Then when you have the primary infection, we have a T-cell selection, we have our T-cell repertoire against this serotype, but in the moment of the secondary infection, we have one clonal expansion of the old T-cell repertoire also, that we have weakly cross-reactive T-cells. Taking together these weak T-cells and the antibody-dependent enhancement in the other side, we have excessive cytokine production that can lead to a cytokine storm and the pathogenesis of the severe dengue. So in this model, the antibody-dependent enhancement, it's always being cited and published. Even now with the coronavirus vaccines, we are seeing a lot of people discussing antibody-dependent enhancement and how it could affect the pathogenesis. A few years ago, we are exactly when the Zika virus was introduced in Americas, we are starting a dengue vaccine project. And this manuscript published at that time, showing that in vitro, antibodies that react to dengue also react to Zika. Antibodies that were able to bind to Zika were promoting ADE. And that, that has been published several years ago, much before that. And that in vitro, these antibodies can generate ADE, but does not mean that this immunity to dengue may drive greater Zika replication, have clear implications for pathogenesis and vaccine programs. I think that's extrapolation, and we have to take a lot of care doing these extrapolations. But what is ADE? So we did a review on that with my colleagues Carlos Sariol and Nikos Vasilakis, and we tried to give some definitions for ADE. So what is ADE in vitro? It's a common experimental phenomenon. We can do ADE in vitro with some, it's not difficult to do. Just give antibodies, a correct cell line, and you play around with antibody concentration. At some point, you can get ADE. We have ADE in vitro really well defined for alpha virus, rabies, Coxsackie, coronavirus, HIV, but there is no evidence of worsening of symptoms or any clinical impact or real world impact of this phenomena described in vitro. But in flavivirus, we can do ADE in vitro easily using heterotypic antibodies. In fact, if you work with homotypic antibodies against dengue under certain concentrations, you can also induce ADE in vitro. You know, other flavivirus like yellow fever, Japanese encephalitis, and even Zika can induce dengue ADE in vitro. But is there any evidence that this really enhances disease? Well, in fact, the only evidence that we have that ADE does occur in vivo, it's in dengue virus, okay? And this ADE is not only ADE, but also this ADE in vivo is associated with a worse clinical outcome, and we can detect in this subject an increase in the viral load, a very dramatic clinical impairment, and a cytokine storm characterized by increase of EL6, 8, 10, interferon gamma, some growth factors, and TNF-alpha, TNF-alpha that induces a poor prognostic outcome. Based on that, we proposed a ADE definition that will be, we propose it as a common experimental in vitro phenomenon, but an uncommon in vivo occurrence that when happens leads to a worsening of the clinical presentation associated with hemodynamic change, increased viremia, and a pro-inflammatory cytokine profile. So this is what we proposed as ADE, but does this Dengue-Zika ADE really exist? What I mean is, if you pre-expose someone to dengue and then infect with Zika, does this ADE happen? In animal models that's been published by Bardella and colleagues in Science in April 2017 showed that in mice model, yes, you can find this enhancement. But in the other side, we look for human subjects in Brazil, and I presented this work many times before. We show that the viral load, clinical symptoms, and cytokine response does not support this hypothesis of ADE in dengue primate Zika infected patients. And in fact, we have the opportunity to collaborate with Dr. Albert Ko, publishing this manuscript in Salvador that was published in the Science in 2019, showing that, in fact, in a population that was exposed to dengue in large scale, instead of enhancement, what we found was a protection for Zika infection in dengue primate subjects. So we could not find evidence of this ADE in dengue primate Zika infected subjects. But what about the microcephaly problem? Because this has been associated to, or at least hypothetically associated with this exposure to dengue. So we looked at, and we published this a few months ago, myself and Dr. Patricia Brazil, look for different regions in Brazil. We look for a region that we have a lot of microcephaly and another city that does not have a lot of microcephaly, but both cities have a Zika outbreak. And in fact, when we look for the dengue titers, they were not even the titer, but the avidity of dengue antibodies. In fact, they do not have they are not a risk factor for microcephaly or other abnormalities. And in fact, when you have heterotypic infections or multiple dengue infections, we have some degree of protection against microcephaly. So over the years, we get to this model that we think works for dengue primary infection, then followed by a secondary dengue infection. You can have AD in vitro, you can have AD in mouse, you can have AD in non-human primates models, and even in human. But in Dengue-Zika, we can do in vitro, we can do in mice model, but as has been published by Dr. L, we could not find this effect in non-human primates. We could not find this effect in humans. But what happened in the other way around? So after we had this, the Zika outbreak in Brazil, we start to see patients that were Zika primate and then later dengue infected. So this can lead to severe forms of dengue. This is a dengue subject from my hospital for a few years ago. And there was something that called our attention in 2019. We have our Zika outbreak in 2016. And 2-3 years later, we have our Dengue 2 outbreak. This was a new lineage that was introduced in Brazil. And we have a clade replacement that is associated to increase of severe cases, but the number of hospitalized subjects, not the number, but the percentage of severe cases of dengue was much higher in 2019 than compared with other years. So we decided to look with this in more detail. So we work with a hypothesis that the previous exposure to Zika would influence the evolution of subsequent episode of dengue. Of course, the new hypothesis that there is no evidence of no influence of the prior exposure to Zika on the evolution of dengue, but our hypothesis there is influence that can be either protective or harmful. So if there is an influence, what mechanisms are involved? So this is what we try to do in the since 2019. Of course, we have a lot of problems with coronavirus during this time. So this is where we study. Our study area, São José do Rio Preto. It's a city in the northeast of São Paulo state, in the central south Brazil. It's a city with roughly 500,000 inhabitants, and it's a really nice place to live if you were a mosquito. We have a really tropical weather with a raining summer and a dry winter, but an average temperature of 23 degrees Celsius and rains a lot. So we have a really nice place to breed mosquitoes. And we have a long history of several arbovirus, different arbovirus circulating in the city. So in this outbreak in 2019, we analyzed almost 3,000 samples of dengue suspected patients. About 1,500 were eligible for this study based on laboratory criteria. I will not go in details, but pretty much laboratory confirmed dengue case by PCR. And about 1,300 have the complete medical record information of that. So when we look this, we have 21 cases of severe dengue, 385 dengue with warning signs, and 800 cases of dengue without warning signs. When we look for these subjects, about 20% of these subjects were naive. They never had either dengue or Zika before. 4% of them were previous exposure to Zika, but not to dengue. 60% of this group was already exposed to dengue before, and about 15% of them were exposed for both viruses. We decided not to analyze this. We decided to analyze naive subjects, Zika primate dengue infected, and dengue primate dengue infected in our study. So when you look this, of course, this goes as usual in Brazil. Our peak of cases are at the beginning of the year, January, February, March. And we have always, when you look for weeks, we always have these two waves. That's something that's really funny because we have the carnival here. So I think that most people don't report or really have a decrease. Then you have the carnival travel, and again, the disease goes up after the carnival. So this is, I'm not going to go into details here, but just to let you know that we have a lot of clinical data from all the subjects. And again, this is, in Brazil, dengue is still a disease of adults, not as a child. So the average age of our subjects is 40 years old. Again, out of the subjects, 812 were dengue, and about 223 were severe dengue or dengue with warning signs. And we have here all the details of what are the warning signs, the severity signs. So we have central nervous system involvement, hemodynamic changes, bleeding, respiratory distress, and several other signs. So when we look for the difference between these two groups, there is no difference in age group, no difference in sex, and no difference in comorbidities. So nothing, this does not explain the differences in between the groups. But when we look for this, for the dengue classification in the naive group, 67% were dengue without warning signs, and 32% were dengue with warning signs of severe dengue. When we look for the dengue primate, we see some protection. In fact, we have 82% of the subjects were dengue without warning signs. But when we look our Zika primate dengue infected, almost 55% of the subjects goes for dengue with warning signs of severe dengue. And of course, this leads to a much higher number of hospitalization in this group. So what I have here, I can tell for you that subjects that were previously exposed to dengue has a more severe, previous exposure to Zika has a more severe disease and get hospitalized in a higher frequency when compared with both dengue primate or naive subjects. When we look for the clinical signs, we saw that in laboratory aspects, only leukopenia was much more important in this group, dengue negative, Zika positive, and also some, the platelet numbers are much lower in this group. So when we calculate the odds for that, so if you assume that the naive subject has an odds ratio of 1 to have a severe disease, previous exposure to Zika gave us an odds ratio of 3. And when you have previous exposure to dengue, actually we have an odds ratio of 0.5, showing some degree of protection against that virus that was circulating at that time. When they look for the risk of hospitalization, again, our odds ratio for the Zika primate subjects are about 3.7, and our for dengue primate subjects about half. So I can tell for you now that during this 2019 dengue outbreak, that was a dengue 2 outbreak, we have some kind of protection by previous exposure to dengue, but we have some kind of enhancement of the severity of the disease when you have Zika primate subject. Since we defined it before that ADE should be defined by a pro-inflammatory cytokine profile, enhancement of the disease, and an increase of the viral load. We decide to look at that. So when we look at the cytokine profile from these subjects, we saw that the dengue subjects, the dengue prime subjects have less interleukin A, they have less interleukin 6 than the naive subjects, showing some kind of protection, less growing factor. But when we look our Zika subjects, they have less interleukin 1 when compared with dengue, but much more interleukin 1 receptor antagonist. That's a soluble receptor of interleukin 1. But also, as expected in the dengue primate subjects, we have, we have increase of growth factors, we have increase of interleukin 17, an increase of interferon gamma for the dengue primate, as expected. I'm so sorry for that. But in Zika primate, we have an increase in EL10. So we don't have a pro-inflammatory response in Zika primate subjects, not a cytokine profile of inflammatory response in Zika primate as we see in dengue primate subjects. And in fact, when you look for the ratio of interferon gamma by EL13, that interleukin 10, that shows some kind of balanced Th1, Th2 response, we have that the Zika subjects have a different Th1, Th2 balance compared with dengue primate and naive subjects, and also has a less intense inflammatory response. So we also decide to look for the viral load of these, comparing these three groups: naive, Zika primate, dengue infected, and dengue secondary infections. And in fact, the only difference that we have is that subjects with secondary dengue, they do have higher titers when compared with control and compared with other groups. There is no difference in viral titers between Zika primate and naive subjects. So we can conclude in this study that there was no difference in the frequency of dengue signs and symptoms according to the serological status. There was a clinically significant difference in the frequency of more severe forms of dengue with warm signs and severe dengue, being these much more frequent in patients with a Zika primate when compared with control. So previous Zika infection was a risk factor for severe forms of dengue and also a risk factor for hospitalization. But the cytokine profile of Zika primate subjects is more towards Th2 response, and dengue primate Zika infected, dengue primate, dengue infected are linked to increased pro-inflammatory cytokines like interferon gamma, IL-17, and others. There is no difference in patients with a history of Zika infection, in viral load, of course, in Zika infection compared to control. Patients with previous dengue infection, they showed a lower risk of severe forms and hospitalization. Yes, so my final conclusion is that there is an enhancement of the disease in subjects previous exposure to Zika, but the cytokine and the viral load aspects does not suggest a classical ADE effect like we see in dengue primate, dengue infected subjects. And of course, when we are finishing this, the group from Leah Katsanik and Eva Harris showed that in their population in Nicaragua, that previous exposure to Zika also was a predictor of a subsequent harmful episode of dengue, but they haven't discussed in details cytokines, viral load, and ADE. So this study was done by now our junior faculty here, Dr. Cassia Stofaletti, and her student Fernanda Dourado, with a collaboration of Alice and Bruno from my lab. And this is the whole group of my lab in the old times that we could congratulate before, concentrate and congregate before the COVID. And I would like to thank my supporters, FAPESP, my main funding agents, NIAID, that's funding this work, also other Brazilian agents, and my collaborators at UTMB, New Mexico State University, MIT, Gorgas Institute, Carey, the Brazilian Army, the Instituto de Medicina Tropical in Manaus, and other federal universities in Brazil. Thanks. This is what I have to say.
Thank you so much, Dr. Nogueira. Thank you so much, Mauricio. That was really a great presentation, very comprehensive, and obviously on major topics and with a lot of broad implications. Thank you again. A very great lecture. So we have, as always, you have the possibility to ask questions on the Q&A thing, and we also had, yeah, we have all the questions here. Can you see them, Mauricio?
Yes, I can see. Do you want that I make the question?
I believe the first one is from Dr. Santolala.
Yeah, he asked us about if the previous dengue or Zika was auto-reported. No, we did this by immunological assays, ELISAs, and neutralization. Okay, so we have a very, we are quite confident in these data. They were not auto-reported, they were tested.
Okay.
The second one is from Matt Collins from Emory University. Matt's asking me the comment of importance of time. Yes, Matt, there is a lot of importance of timing, because we know that when you have you have the infection, you have high levels of antibodies, and we're going to have a cross reaction that neutralizes. Over the time, this goes down to, and going to get a range that you're going to have ADE, and that's going to go on time. And after that, nothing. Of course, if the same outbreak had occurred 1 year before or 1 year later, we may have different results on this data, because the timing is crucial for that. The next one is from Paola. Paola is from Pasteur Institute in São Paulo, University of São Paulo. Paola, thanks, Paola. Does the antibody involved in ADE are low affinity antibodies that can recognize multiple antigens without a specific receptor? Can one think that these antibodies are just results of polyclonal B cell activation? Paola, we try to play around a little bit with affinity in some aspects, and we couldn't find anything regarding that. There is no direct link on that. That's a very complex aspect of the biology, and I think that we need to move a little bit better on that. But these antibodies are results for a polyclonal B cell activation that activates the B cells that were there were memory cells from the previous infection. So we have antibodies that are specific, antibodies that are not specific because they weakly cross-reacted. And I don't know if avidity or affinity, that they are not the same thing, may be involved, but we need to measure this better. And I think that these are these questions. There is.
We also had, there was a question on the chat, if I'm correct.
Yes, correct.
From Dr. Glenn Richard. did you see it? Does previous dengue infection protect against increased morbidity induced by a previous Zika infection? That is the fourth group on what you presented.
No, the fourth group, Dr. Richard, is that were subjects that were previously exposed to both dengue and Zika. And we decided not to, because we cannot first what have mixed analysis. No, we cannot say that nothing about the fourth group.
Thank you. And we have another question now from Dr. Alan Schmadjon. Do you see it?
Hold on. How does the type-specific and protective role of anti-NS1 antibodies influence your thing? We haven't measured that, so I will not guess. Okay, I would say, I would be quite honest and say, I don't know. I need to think about it. And thanks for the question. That's something that now I need to think about it. That's the beauty of science.
Thank you. I believe that those were the questions. I have a comment which is very general, and actually you have mentioned this in your presentation. But the comment on the impact of ADE for coronavirus infection. See, so you did mention that there is not such evidence from your experience, but just, you see, this is an example for me of what the GVN is about, because at the very beginning, during the summer 2020, when the first vaccines were being discussed against SARS-CoV-2, I remember a lot of discussion within the GVN COVID-19 task force about this possibility, you see, of ADE and how, and this is really how the cross discussions, I would say, between different fields of virology within the GVN can be very useful. And this expertise that you have mentioned on this topic is important, and I believe still relevant for the development of further vaccines. So I do not see other questions. So, well, thank you very much. That was really a great lecture. And again, which very well introduced the dengue task force that we are initiating. And it will be a pleasure to work with all of you on these very important objectives. The GVN obviously has been very much focused on COVID-19 for obvious reasons, and will still be very much focused on this. But we are working on all human viruses, and we had today an excellent example. Thank you so much, Mauricio.
Thank you very much. It was my pleasure.